We move to the next presentation by Michael Pfaffel. I'm just wondering if it's really necessary to introduce Michael, because I'm sure that people interested in qPCR and gene expression are all know him. He's one of the leading scientists in the field of gene expression and gene quantification. Do you know that his paper, a new mathematical model for relative quantification in real-time PCR, published 22 years ago in Nucleic Acid Research, has been cited today nearly 22,000 times. That means 1,000 citation by a year. Impressive. Today, he has reached the principal investigator status at the Technical University of Munich in the Institute of Animal Physiology and Immunology. And I have to add that he is the initiator and lead organizer of the qPCR, dPCR, and NGS gene quantification event series in Freising since 2004. Yeah, 2004. And today is going to talk about the development of analysis pipeline for microRNA biomarker signatures in molecular diagnostics based on circulating EVs. Thank you.
Thank you, Patrice, for this very nice introduction. Thank you for staying here, and everything worked well now with the presentation. Thank you very much. So we are working on biomarker development. We started with PCR long ago, as you heard, in the late 90s. So we were one of the first buying the light cycler, and then we worked a lot with rotogene. But from the PCR, we went over to the next generation sequencing, and from the classical mRNA quantification, we went over to microRNA quantification, and then of course we hit vesicles at some way. And I want to tell you now our strategy. We have lots of collaboration, how we do that and how this was done the last 10 years, more or less. So, but the first most important thing is, do we have a free or cell-free RNA in our cells? And as seen here in a nice Nature Biotechnology paper, there's lots of cells and lots of vesicles in our circulation. And if you go deeper, you can allocate these RNA, as seen as well in your talk, to a lot of tissues. The majority, as also stated by Peter, in blood, here is blood as an example, is coming from the blood cells itself. But of course, you can find as well RNA from all the tissue, because blood is circulating and taking up RNA from any kind of tissue on the way. The big question is, what is the function of the so-called extracellular RNA? And we know this, that the extracellular RNA is a kind of communicator, cell-to-cell communication via extracellular RNA, and of course proteins and so more. And but how these RNA, how does it really working, and how the RNA is delivered? And there are some appointments, that it's intrinsically stable in the blood and in the urine. This was the first meaning. No, this is definitely not true. Therefore, the RNA need to be stabilized, need to be bound by proteins, or need to be bound by vesicles. That's what we know so far. And since it's bound, it's stable, it cannot be degraded by RNases, which are in the circulation. And we have different kind of vesicles. I come to that in the next slide. But then the idea was also born about last year or the last years, that the EVs have also kind of a corona, a protecting area around the EVs, and they bind kind of anything, in my opinion. They bind RNA, DNA, what else, proteins, and this corona story we are following quite now. So we are applying first different isolation of EVs, rigid, rough methods where you have really pure EVs, naked EVs, you also can name them. And on the other hand, you have kind of crude EVs with a corona around, and they have more RNA content. And therefore, we want to follow how these isolation methods behave. And what we also know that these good and naked EVs, highly isolated EVs, have no biological function because the corona, more or less, is interacting with the target cells. Okay, so, but we are investigating this. How is the effect? How much corona do I need? And corona also means contaminants because there are proteins bound, but also, of course, nucleic acids. So the story is we want to using liquid biopsy to more or less have access to the circulating extracellular RNA, and we use these for biomarker discovery. And as you see here, you have a different liquid biopsies, of course, blood, urine, saliva, milk, stool. We heard a lot about that as well in Peter's talk, and we are focusing here more or less on the microRNA or in the wider range on any kind of RNA in connection to EVs, or on this slide it's named exosome. So this is our focus. We want to characterize this and then draw our conclusion for biomarker, for prognosis, for different kind of clinical models. That's our goal. So if we look into the cell, we have a multitude of different vesicles in the cell. We also heard this today, and the big question for us, or the first question around 8-10 years ago, what is the best source for RNA biomarker discovery? Is it more or less the full blood or the packaging blood, or however you stabilize it? Are these the white blood cells itself? Is it the cell-free compartment? Could be cell-free RNA, cell-free DNA, or is it connected to any kind of vesicles? And if we are talking about vesicles, the next story is open. Should we use exosomes, the microvesicles, the ectosomes, and so on, or the whole community of all the EVs? And therefore, our first study was which compartment is the most important or the most informative for our studies. Therefore, we compared different blood compartments from one and the same patient, cells, serum, and the EV compartment. And since we are collaborating with clinics and we have sepsis more or less in the focus, we compared 10 sepsis patients, isolated 1 microliter serum, packaging blood, and the EVs from 1 microliter. And just jumping over directly to the results, you can see different compartments have different read counts in the tissue, and you clearly see here the most is definitely in the packaging because you have the entire blood. And if you have here serum, or we named it these old days exosomes, but if you zoom it up, you clearly see that here in the packaging and also in the EV compartment, you have a kind of enrichment in comparison to the serum. So any kind of EV pre-isolation for after RNA sequencing might be an enrichment. This was our conclusion. That's why we went for these EVs. And just to show some examples from this first preliminary study, so same patients and everything, then a sequencing, you see here the cells have their own signature in terms of comparing 10 healthy with 10 sepsis patients, and the serum and the exosomes, they share more or less the same because the EVs and the exosomes are part of the serum, but of course in the exosomes they are enriched. And today we can say it's about enrichment 3 to 5% if you do a crude EV enrichment before your sequencing. So this brought us to the advantages of the EVs. Therefore, what are the advantages first of the microRNAs? We have a liquid biopsy, which is non-invasive. We have microRNA as short targets. That's why they're stable. We've proven this long ago. A microRNA 21, 22 is much more stable compared to mRNA, which is an average close to 2,000 bases. And the microRNA are in or at the EVs, meaning they are protected. The next thing, we are talking about microRNA expression changes. This allows an early and easy detection of these biomarkers, and we can work with a signature. And a signature is always more stable compared to a single biomarker because it's more stable, it's standing on multiple feet, and the signature is more stable. That's it. And so the advantages of these signatures are it's a specific information depending on a disease, so the combination of 10, 15, 20 microRNA, and we can directly rely to a certain disease or a cancer type or status. And if we have these microRNAs identified, these marker signature, we can easily apply PCR and amplify this signal. First, we do the screening via next generation sequencing, then we develop a signature, and then we put it in the PCR cycler, and then we can easily apply it. That's why we have also a high abundance of the signal, because we can have PCR cycles in between. That's why all sounds very easy, but we have to standardize it, and we're also working on that. And since we are part of the MIKE guidelines and part of the MICEF guidelines, we have two guidelines which we have to follow, and I will show it how you should make it. That means all looks easy, but as usual in the lab, you have your outliers and where the variability is coming from. Normally it comes always from the very beginning. You showed it also very nice. The sampling, the storage, the experimental design. This introduces a lot of variability. Of course, the isolation, the characterization, the RNA extraction, the sequencing itself, and of course, at the end, the bioinformatical analysis, okay? The bioinformatical analysis can a little bit correct for this and that. This will be shown later by Benedict, but in the beginning, you have to do everything right to get good results. So, coming back to the validation, possibly if you're working with EVs, you heard about the MICEF guidelines. We also contributed to the MICEF guidelines, and they were developed long ago by Jan Lotwal, and then they were taken over by Ken Witwer and Clotilde Thury. And I think we are now on the way to publish the update for 23, and these guidelines show you how you should work with EVs to be reliable. You know it maybe from the MIKE guidelines. This is more or less the same. One is for PCR, the one is for MICEF, and it was kind of a copy and paste of the MIKE guidelines. So the nomenclature, the collection and pre-processing of the EVs is very important. Then how you separate them and how you concentrate them. And of course, the characterization of the EV is a central part. Do I really have EVs, or are these aggregates of proteins or bubbles or whatever? Good. So then we started to establish a MICEF compliant EV workflow. Sounds great, okay? But it's not that easy. So we optimized different EV isolation methods. This study we started about 16, 17, it was published 18, and during this day, the goal was it must be fast and reproducible, since it has to be done in the daily clinical routine, and it should be suitable for RNA biomarkers, because we are working on RNA biomarkers. Okay, and what we did, to make a long story short, this is a paper from a former PhD student, Dominique Buschmann. We compared different isolation methods, ultra-centrifugation, mercury, exospin, QED, exoRNA. These were the methods available 2016-17, and then we characterized all from the same patient, all done in parallel, all in 1 milliliter, and then we have our suspension, and then we characterize them by Western blot, nanotracking analysis, and electron microscopy. And then we compared these different kits on the basis of the RNA results, meaning we've done one big sequencing approach, all in parallel, and from the different expressed microRNAs, we draw our conclusion, what is better and what is worse. Then, this was kind of the result, first crude result. We compared all the reads, and you clearly see some methods clustered together. So we have two big clusters like exospin and QED, which is a little bit mixed up. and then other isolation methods like mercury or exo-RNA or differential ultracentrifugation, they cluster quite nicely together. Here, again, these are the sepsis patients, the volunteers, and so on. And the first conclusion from that, yes, there is a huge impact on the EV isolation of your later RNA signature. And I can conclude it, if you join the workshop, you've seen the sizes are different, and the distributions are different, and. And based on this, we went over to the significantly regulated genes. And of course, if you have a good isolation and you have a huge output, and if you have a huge output on RNAs, you have more candidates, possible microRNA candidates, which you can implement in your bioinformatics to find a stable biomarker signature. And very interesting, only 19 microRNA are shared between all these isolation methods. And here you have really some outstanding here, like mercury or exo-RNA, which have a lot of individual biomarkers. Based on that, based on MIKE guidelines, always validate yourself. So we developed the biomarker signature, and also the highly up, the 10 most up and 10 most down regulated. We have to validate, because it's actually the reason. We're looking for biomarkers, but the real assay should run in the PCR. Therefore, you have to amplify it into PCR, and then we have our kind of threshold levels. At least 50 or 70 reads are good to can quantify it in PCR. Better is if you have 500 or 1,000 reads, that is more stable. So we played around with the candidates that you can kind of confirm and validate them. And here you see one of these first validations, up and down regulated. Definitely you end up with different results coming from NGS and from PCR, but they look kind of good, and this was a kind of validation for us. We could validate them and re-amplify them. And based on these biomarkers, we then compared in this PCA as a different isolation method, and you clearly see here the isolation method, the isolation method with the most candidates gave a clear separation of the clusters. Here you see 10 patients, here you see the 10 patients. And here the other methods which delivered less biomarkers have not a good separation. But, I have to say, but you could separate the healthy from the sepsis patients with all the isolation methods, okay? But some are better and some are not. This was published in JEF 18. So now we have our status, we're using EV, we know how to purify them, then let's start with the study. And then the clinicians already had sampled lots of samples, so we decided to make a first training cohort in sepsis patients. We classified our biomarkers, then of course we need a functional and physical validation, and then we had a second cohort, independent, and we looked up if our biomarker are also working in the other patients. And this brings us to kind of the workflow. This is kind of the central picture of this talk on the workflow. So we started here with biological samples, and at the end we need a kind of biomarker candidate pattern. So we started here with the first cohort. These were 67 patients. First, you do the EV isolation, the RNA extraction, the small library prep, the sequencing, and of course, then the data analysis. And in between, always, we had kind of control steps. Of course, here, EV-qC, MySIF guidelines. Here, RNA-qC, micro guidelines, RIN values, and something like that, okay? Then library prep, this is already involved in the bioinformatics. How good is your library prep? How good are your NGS data? And then, of course, if we have these biomarkers, we have to validate our candidates via PCR. And then we generated a kind of first candidates in the training cohort. And this has then further on confirmed in independent patients, another 75 patients, is it really true what we found before? Okay, here's some data. So we compared in the beginning much more sepsis studies, clinical cases, but at the end we fused it, shrink it down to 3 groups, to volunteers, to healthy, community-acquired pneumonia, also early level of sepsis, and real sepsis cases. And at the end, it turned out that we could separate these groups with 12 microRNAs, okay? And then, of course, we have to validate these microRNAs via qPCR, and this is this slide here. You see this is the training cohort, the validation cohort. We did NGS in the training cohort, we did PCR in the training cohort, and only, of course, RT-qPCR in the validation cohort, and more or less about 70 to 80% in concordance between the different patient groups. So we saw that we are valid, we could reuse our biomarker signature, and we could also classify the patients. And this is the classification of the patients. In the training cohort, we could derive with these 12 microRNA an accuracy between 92 and 99%, depending what you're comparing. And then later on, in the prediction accuracy for community-acquired pneumonia and sepsis, we are in the range of around 80%. And I actually forgot it. These are all clinical samples drawn from the first day when the patient came to the clinics. So it's not over the sepsis. So the patients are coming to the clinic, the blood is drawn, and then on the first day at ACU are all of these samples. We have also further data, but in this study, we just implemented day 1 at ACU. This was also published. And then we even went further on. We said, oh, 12 microRNAs, this is too much. And then the bioinformaticians, they played around, and then we fused both patient cohorts. So at the end, we had then 152. And based on these 152, of course, if you have more patients, the biomarker are slightly changing. And then we could even shrink it down to these 3 microRNAs. So 193A, 542-3P, and 1246. And these 3 are kind of a marker for community-acquired pneumonia. And to make it even more simple, we just calculated the average of the crossing point and plotted it as a kind of very small biomarker signature. And you see here, it's highly significant, okay? So, looked good so far. And then I had a little bit to announce the talk in the afternoon. Of course, all these samples over 10 years or even 12, I would say, Benedict developed a biomarker platform, which of course helped doing all these analyses and make it as easy as possible. So what we've done so far is kind of doing small RNAs. Now we enlarge it to longer RNAs. We started as well with long non-coding RNAs, of course, also messenger RNAs, which are connected to vesicles. Then, of course, all the statistical methods, multivariate analysis, and then also physical and in silico validation of all the microRNAs. But this will be then later on introduced by Benedict. This caRNAge pipeline. We also developed so-called a MyRef, a tool, because in PCR, and if you're working with EVs, you need normalizers according to the microRNA. Where the microRNA normalizers come from? And I have to tell, especially the guys from QIAGEN, these normalizers in these microRNAs are not microRNAs. Therefore, you should change a little bit the strategy. You have these, what do you have? Forgot the name, but these are not microRNAs, which are definitely in these microRNA kits. And then also we are focusing further on to find more biomarkers. So the isomers are in the center as well. So we are losing a lot of information. We're just looking on the canonical. And if we implement microRNAs, we could even double or triple our counts for a specific kind of microRNA family. So Benedict will show this later on. And then the pipeline is full. All RNA classes are analyzed, so you can select whatever you want and input in your analysis. I will skip this slide because this is definitely in Dana's talk, but we have isomers, we have differential gene expression, we have functional validation. It's a big tool which should help you to analyze your RNA sequencing reads. Isomers, tRNA halves are also inside, and I will skip these slides since we are late, and as well the MyRef, which is also implemented and all published so far. So, coming to my last slide, what we've done, or what is, first you have a group, you do the liquid biopsy collection, then you have kind of integrated omics. For now we only have RNA, but in the future we will get definitely more. So we have here a first patient cohort, a training set. We started as well with proteomics. We want to implement as well mRNA and long non-coding RNA, but this is a little bit complicated, the long non-coding RNA. And then the second is more or less a biomarker validation in independent cohorts. And maybe, if we are lucky, we can implement our results as well in the clinical testing. And with that slide, I want to thank my collaborators, or our collaborators, mainly from TUM and from LMU in Munich, some companies and our funders, and as well the people who really did the work with the programming, with the bioinformatical pipeline. First row, it's Benedict, and also Alex and Esther were involved. And with this last slide, I want to say thank you as well to other collaboration partners, which projects we have with all of them together. And thank you for your attention, and I'm very happy for any kind of questions. Thank you. small-RNA, or was it small-RNA?
I just have to ask on your comment, because the mercury control set is 3 synthetic microRNAs plus the C. elegans microRNA, and there is another one that is spotted on the panels. We did use U6, but that's 15 years ago. Yeah. Okay, yeah, sorry, maybe.
Okay, sorry.
Nice, we could clear it up.
Michael, I was wondering about the unmapped reads. You showed a plot where you had quite a bit of unmapped reads in the small RNA data.
Maybe will answer this later on. But we could reduce the unmapped to I think, to a minimum. Yeah. But smaller than 16, or? The question was about the unmapped reads.
Yeah, I'm not going to talk about it later. We usually encounter quite a few number of unmapped reads in this case, but it only depends. Unmapped in this case is always in relation to what you're using for mapping reference. So if you're going for a transcriptomic approach, and whenever you have any RNA that belongs to the intro, you also have unmapped because it's not on the transcriptomic reference.
Okay, so it's unmapped relative to a transcriptome, not to a genome.
Yeah.
Okay.
You have it on the genome. You but in this case, it would be chronic.
Okay.
And it could also be I don't know.
I was just wondering if there was a possibility that these were bacterial RNAs that would also be in the. Okay.
In the case of that study, definitely now also study that specifically looking for bacterial RNA. So yeah.
Yeah, I have a general question. So we have seen so far a lot of people isolating extracellular vesicle from plasma. Here we have an example from serum. Do you have a general recommendation? Are people using more plasma because it's easier?
I think every method has its advantage and disadvantage. We're also working in urine and something like that. We have our own patents with glycan-based isolation. Everybody has it. Every method has its advantages and disadvantages. In some it's easier going, you have more contaminants, and we also tried combinations, in terms of purity, using 2 physical characteristics, size and density or something to get better run. But I would say this topic is totally will be discussed totally new when we know what's the impact of the corona. But then only the functional will be of valid. Not for us. For us, biomarker is fine. It can be crude, it can be anything inside. We can pick the biomarkers via PCR, it's specific enough, fine. But for kind of for functional relevance, then the isolation method, like TFF, tangential flow, and something like that, will be in the focus, will be more in the focus. Continuous isolation of biological active EVs, yeah. I think we have to discuss this in 2 to 5 years again, yeah. For now, the goal was to make it as pure as possible, as naked as possible. And then it turned out pure is fine, naked is fine, but it's not working. Oh, go back and make it biologically active.
Thank you.
Welcome.
Thank you. No more questions? Okay, so we move to the next presentation. Thank you.
Thank you.